Hi there, welcome. We're going to start our session. Let me see if I can. So this is the Phages, the Rage, Demystifying Phage Therapy. Can't take credit for this title, but I hope we can learn quite a bit from our colleagues about phage therapy, and its relevance for the CF community. I'm John Koff from Yale University. Isaac Martin is here. We're going to co-chair, and we are told to remind you that we're live streaming this, so if you're going to ask questions, please come to the microphones. Introduce yourself, because it's really hard for us to see with the glare behind, and ask your questions. Questions are also available on the app if you wanted to do so, and we'll obviously read those questions. So we have no disclosures. So just to start off with, we're going to be talking about bacteriophages or phages. So these are viruses that specifically infect bacteria, originally discovered in 1914, kind of identified by Dr. Twort in England and Felix Jarrell in 1917. You can see a picture of Dr. Jarrell on the right. He's got a Yale connection, spent some time on faculty at Yale, and we've got records in our library about him, so that's kind of neat for us. In terms of phages, these are ubiquitous in the environment, predicted 10 to 31 phages, so more phages on the planet than any other organism, and they outstrip bacteria about 10 to 1, which would suggest that we can find phages for the bacteria of interest that we want to use to potentially treat patients. I've put a figure here of a EM diagram of a classic phage, and then the phage inserting genetic material into the bacteria. And what I'm showing you here is the lytic life cycle, where the phage will bind to a receptor on the surface of a bacterial cell, introduce the genetic material, and go through the lytic life cycle. This is important for us to consider for human phage therapy, because we're only going to be using lytic viruses that will result in destruction of the bacteria in the context of phage therapy. Interesting characteristics for the phages are that they have the potential to degrade biofilm, which is obviously relevant to the cystic fibrosis lung. There's a potential for quite a bit of specificity. Individual phages typically interact with specific bacteria, and so this offers the potential to be quite selective, which is interesting when we think about our treatment options. And currently, pharmaceutical development has required the use of kind of a cocktail approach or genetic alteration, because these are environmentally available, organisms, and so intellectual property requires that approach. And I think that has some significance in terms of how we've thought about using phage therapy. So just to summarize a nice paper from 2023, if you're interested, this is a summary of existing publications for phage therapy. And in the upper right, you're looking at case reports since 2000, and pulmonary infections were the number 2 indication for those types of reports. Obviously biased by the potential for only for folks to report successful cases. And so that's an important consideration when you think about the existing literature. And on the bottom, you see a really wonderful increase in the number of clinical trials that have been initiated over the course of the last several years. So this is very exciting for us in this community. And so we've got our speakers here. We'll be introducing them, but I'm going to let Isaac introduce himself and talk to you about a project he's working on.
Great. Thank you so much, John. Just let me navigate to my... So good morning, everybody, and welcome to our session. It's my great pleasure and honor to be here amongst very distinguished speakers in the field. I'm a pediatric respiratory physician in Toronto, Canada at Sick Kids Hospital, and I will be speaking to you about a small project. So I'm going from more theoretical, I've just got funding for this, so we're just starting, to some of the more clinical things that my colleagues who you'll hear speak later in the session. And I feel like, at the risk of remystifying phage therapy, I don't have anything to disclose. So the learning outcomes for my presentation, I hope that by the end you'll be able to describe some key anti-phage defense systems in Pseudomonas aeruginosa and their implications for therapeutic phage susceptibility, and maybe explain how bacterial genomics underpins precision approaches to phage therapy design and future trial development. And in this, recognize how basic and translational research intersect to shape the current clinical phage landscape. So I think if you take nothing else from my presentation, which at moments will be a bit technical, it's just that kind of setting the stage for the other speakers, in the sense that for as long as bacteria have existed, they've been in constant conflict with the viruses that infect them. These are bacteriophages, or phages, which have been nicely introduced by John a second ago. And this co-evolutionary arms race has been shaping bacterial genomes for billions of years, driving the development of highly sophisticated defense systems and equally sophisticated viral counter strategies. Each bacterial lineage carries within its genome signatures of these battles, systems that you may have heard of, like restriction modification, CRISPR-Cas, and abortive infection, all designed to recognize and neutralize invading phages. In turn, phages have counter-evolved mechanisms to evade or suppress these defenses, like anti-CRISPR proteins or genome modifications that allow them to escape restriction modification. So I feel, and my co-PI, Dr. Trost, also a recent hire at Sick Kids, feel that this ancient interplay isn't just of evolutionary interest. It directly affects how bacteria respond when we try to use phages therapeutically. Understanding these defense systems is essential if we want to predict which phages will succeed, which will fail, and how perhaps best to design phage cocktails or engineered therapies in the future. My work focuses on mapping these anti-phage defense systems in Pseudomonas from people with CF. And the hope is that by characterizing the bacterial side of the equation, we can begin to understand the genomic landscape that underpins phage susceptibility, and this could lay important foundations for future therapies and clinical trials. So I'm moving from a very fun cartoon-based slide to a very unfun technical slide. And I don't want you to get too wrapped up in this, but suffice it to say that there are many, this is a very burgeoning area in phage and anti-phage defense system research. And I'd really, if you are interested in this, I would really recommend this paper, which was a review in Nature in 2023. And just that there's various mechanisms that phages and bacteria have sort of, as I was saying, co-evolved through. Bacteria can escape, or the phage can either escape detection by the bacterial cell, interrupt signal transduction, and by overcoming phage effector activity. And I think when I think about these different mechanisms, and once again, I don't want you to spend too much time picking apart the individual details of this slide, but I think about two sort of broad categories. So one category is mechanisms evading phage. So the bacteria recognizes that nucleic acids or nucleic acid sensing mechanisms. And these are very familiar to a lot of people, I imagine, in the audience. They involve restriction modification systems, which cut foreign DNA, lacking host methylation marks, and others like CRISPR-Cas, which provide adaptive immunity by integrating fragments of phage DNA into the bacterial genome and using them for targeted cleavage during subsequent infections. Beyond these, there are dozens of newly described systems that act in similar ways. I won't name them. Some of them are on this slide. And each represent different signatures of non-self DNA or RNA, and then trigger a cascade of responses involving nucleic acid cleavage, degradation, or modification. In essence, these are like, you could think of them as molecular trap wires to scan for foreign nucleic acids and then rapidly neutralizing them before the infection progresses. The second category is phage protein sensing systems, and this second major category, as I sort of described, detects phage proteins rather than nucleic acids, and these usually result in abortive infections. So instead of preventing entry, once the phage has entered the cell, and the bacterial respond once the phage begins to express its own proteins, which often triggers a cellular suicide response that sacrifices the individual cell and then protects, thereby limiting the infection and protecting the rest of the community. One such example is the CBAS system, and I feel like every time I go to the literature, there are new systems described. They all got very funny names. And yeah, I think just understanding that there are different sorts of systems that are involved that we might be able to scan for. And in this same article, they describe very nicely, I think they look at somewhere in the range of 20 or 25,000 bacterial genomes. And in panel A, you can see the average amount of anti-phage defense systems that they find per bacteria. There's a gray bar there, I think I can show you here, which represents the average, which is about 5. There are outliers. I think they found one bacteria, not Pseudomonas, thankfully, where there were like 57 different anti-phage defense systems. You can see the average is about 5. And then in the right-hand panel B, you can see the relative prevalence of these defense systems for the different bacterial phyla. And you can see, for example, that things like restriction modification are extremely common, CRISPR-Cas extremely common, CBAS less so, but then there's other systems that are much, much less common. So our hypothesis is that this is relevant when we're proposing phage therapy or developing phage therapy or clinical trials, and we hope that mapping bacterial antiphage immune systems will enable prediction of phage susceptibility and then guide the rational design of phage cocktails or phage therapy for CF. So we've named our project ARMOR-CF, Antiphage Resistance Mapping in Pseudomonas for Optimized Response, where we'll look at whole genome analysis of a very well categorized clinical isolates from CF patients. We've got a comprehensive annotation and bacterial anti-defense system pipeline. We'd like to do so the first part is very descriptive, describing early transient versus chronic or persistent isolates integration, and then integrating that with phage susceptibility and clinical metadata. So this is a summary of our workflow. Thankfully, we've got a large sputum biobank with CF airway cultures. Whole genome assembly, we've got Center for Computational Medicine, we've created an annotation pipeline, and then, as I said, the first part is descriptive, so doing a comparative analysis of which defense systems are present or absent, what the patterns are, if there's co-occurrence of different systems, and the diversity of these, with the idea that eventually we'll be able to integrate this with some clinical variables and phage susceptibility data. So currently we've sequenced over 1,000 Pseudomonas aeruginosa genomes and assembled these. The vast majority of these are from CF isolates, chronic and new acquisition, but we also do have a handful of isolates that were from environmental sources. We also have a handful that were from other clinical infections that we see in the hospital, like ventilator-acquired pneumonia on the ICU, and bloodborne infections with Pseudomonas as well. And our annotation pipeline has been completed, and we are now getting tons and tons of data that we now need to sift through and make sense of. What we hope to achieve is to identify the defense signatures that are linked potentially to phage resistance or to the emergence of phage resistance. It would be nice if we could see patterns distinguishing early versus chronic isolates. It would be nice if this could inform the selection for therapeutic matching. This is the dream. And supporting rational cocktail design and engineering approaches while laying the groundwork for predictive models. The next phage, which we applied for funding for, is sort of phage adjacent, but using the knowledge that we gain from this part of the trial is to leverage these endogenous CRISPR-Cas systems as therapeutic tools. We hope to drive targeted either self-destruction of the cell, or if that fails, restore antibiotic sensitivity. This has been described in a recent publication from 2019 and seems to be a promising sort of methodology. I say phage adjacent because we've actually decided that we're probably going to be using CRISPR plasmids, but you could also use phage for administration of this. And I think if all of this sounds very sci-fi and very complex to you, I would just like to point you to, I got an alert about a month ago for a preprint of this group in Stanford who actually they've developed their own AI-guided, essentially they've constructed phage that have the ability to infect in live cells. So I think there's a lot burgeoning here as a potential for therapeutic trials coming up, but also with the reality of the fact that this is very early. We don't know what the scope, the safety, manufacture of these compounds will be, but it's a very exciting and very promising field to be involved in at the moment. So I'd just like to take the opportunity to thank first and foremost Chantal Trost, who's a lot of the brains behind this. She's in the genome segment, a recent hire at SickKids, and having access to her lab has been great. Also, Val Waters, one of my mentors at SickKids, who's been very helpful in help setting up this project. The Genetic Analysis Facility and Center for Computational Medicine has been really instrumental in a lot of this, our annotation pipelines and everything. But also, we couldn't do this without the help of the CF clinical research team who collect the samples and put them in the Sputum Biobank. So many thanks for your attention. We've got some time for questions, but also if there are other questions that you forget at the moment, we'll be having a little bit of a panel discussion at the end, so you don't have to feel pressed to ask them now.
Well, I think one question that in terms of your data set that everyone would find interesting is, do you have pre and post modulator samples and longitudinal samples for patients with the genomes?
Yeah, so that's a very good question. We do have pre and post. I don't know yet. I haven't gone through the data with a sort of fine-tooth comb enough to tell you how much pre-modulator, for instance, and post-modulator. But yeah, that's definitely another interesting question that we'll hopefully be able to answer. I see that there's a couple of people at the microphone back there.
Hi, Dr. Martin. Thank you very much for the talk. I thought it was really fascinating. I'm Anna. I'm an MD-PhD student in Von Cooper and Daria Van Tyne's lab at Pitt. And I was wondering if your annotation pipeline is available for the public to use, or if you're still just using it internally, because I would love to investigate more about our own bacteria's phage defense systems.
That's an interesting and very timely question. So at the moment, it's not, just because we're still refining things. So at the sort of stage that we're at, I think it's like almost ready to be finalized. So we've gone through several iterations. There have been a few hiccups along the way. The assembly, the sequencing and assembly bit was probably the easiest part, but then there's a lot that's not known about these, and it's like a really rapidly expanding area. So it will be, the genomes themselves are on NCBI database and they're publicly available. The pipeline is not yet. At some point it will be published. So this will be a publicly available pipeline, but we're not quite at that stage yet.
Okay, totally understandable. Thank you very much.
Thanks.
Hi, James Gurney from Georgia State. Previously, when we've looked for CRISPR-Cas in Pseudomonas after phage therapy, we've never found it being active. Is there any reason to suspect any of the defense systems would be more of an issue for phage therapy?
So you said that when you've looked at this in like clinically applied phage, we haven't found CRISPR-Cas as like a...
As far as I'm aware, it hasn't been shown to be ever active in CF patients.
Yeah, I mean, I think it's interesting. There is some data. So I think this is a very sort of basic science at first principles look at things. I think some Pseudomonas doesn't actually have any CRISPR systems. And actually, I think part of it is just being able to quantify of our CF isolates, which ones do and which ones don't. And maybe is there a connection there, which we don't know, between which therapies succeed and which therapies fail. But I think with looking at the prevalence of some of these other systems, like restriction modification or abortive infection, I think that there's a host of reasons it probably doesn't boil down to just a CRISPR-Cas sort of system. So I think this stuff is very, I guess, scientific first principle, but I do hope and I do think that there will be a clinical tie-in with the data that we get that could be quite interesting and informative for future trials and therapies. Thanks.
Hey, thanks. That was a nice talk. My question is, do you have an idea about the relative importance of phage defense systems versus receptor heterogeneity or the lack of binding, due to, say, for example, population heterogeneity of the PA that infects CF lungs as the mechanisms of poor efficacy of phage? I mean, what's more important? Is it defense once phage gets inside the cell, or is it lack of available receptors and the heterogeneity there?
I think that's an amazing question, and I think it boils down to we don't know yet. But I don't think we've really looked at these defense systems as being I think it's been much easier to look at receptors and to look at just doing a plaque assay and seeing like, oh, now the phage isn't infecting and lysing the cells, or now it's less potent than it was before. I think those phenotypic things have been much easier and but still difficult to tie into clinical outcomes, right? So I think this is going to be but one of the pieces of the puzzle. But I think that's a great question, and it's one that we hope to eventually be able to answer.
Thanks.
Hi, Isaac. Excellent talk. I'm Gail Stanley from the Yale University. In your data set, are you going to include whether or not these isolates came from patients that have received phage or what antibiotics they're currently receiving?
Yeah, so that's also a very important question for the sort of more clinical tie-ins or clinical analysis that we hope to perform after the characterization. We do have that data. To my knowledge, I don't think we have anyone in our data set who has had phage therapy. But interestingly, phages are in the environment, so people will be, even if we haven't administered it as a therapy, the idea is that people will be exposed to naturally occurring phage in the environment, and this will maybe exert a selective pressure on the communities in the lungs. I didn't talk about like lysogens and stuff, but looking at a lot of Pseudomonas DNA from clinical isolates, there's a lot of phage DNA in these genomes. So yeah, linking that and eventually to like clinical things like what antibiotics they're on, what other therapies they've been on, I think is a really important question.
Thank you.
Thanks. Thanks.
All right. So our next speaker is Jerry Nick from National Jewish, and let's see if I can get yours up. Is this you here? That doesn't look great, huh?
Yeah, I think that's right.
There we go.
Yeah.
All right, you're all set. Did it come up?
That's staying. Well, very good. Thanks so much for this invitation to be here and to speak at our very first Phage symposium. I don't have any conflicts to disclose, but I would like to acknowledge my funding sources, especially the Cystic Fibrosis Foundation. So here we are with our newest and our oldest CF therapy. We've heard a little bit about Frederick Twort and Felix de Herel. But what about the clinical trials? Where, after 106 years, where are the clinical trials? And so it all started with a successful treatment of a child by de Herel, who was at the Pasteur Institute as a unpaid volunteer. And he had encountered a boy with severe dysentery, created a flask full of phage, and enticed him to swallow it. He succeeded in curing the dysentery. It was a marvelous response. But in the spirit of the age, de Herel and his 3 interns, they all drank it ahead of time to prove that it was safe. And so that was I guess the first phase 1 trial. And then phage just exploded, and really clinical trials were sort of forgotten. In the 1920s, phage was used extensively to treat cholera, plague, all sorts of dysentery, conjunctivitis. Whole villages were treated. And when it worked, it really worked. In 1925, Sinclair Lewis published the novel Aerosmith, and one of the subplots is that the protagonist is a physician that's wrestling between the use of phage for a clinical trial versus compassionate care. It went on to win the Pulitzer Prize, and Lewis won the Nobel Prize, but it was the Nobel Prize for literature, not for medicine. And then so by the early 30s, phage was available in the US. Eli Lilly had 7 phage products that were available, and it's fair to say that phage sort of deteriorated into kind of quackery. It was kind of used for whatever ails you, and subsequently medical journals, including JAMA, published a series of highly critical reviews about phage therapy, specifically the lack of clinical trials, the lack of controls, the possibility of confounders, and the purity of the lysates, which were predominantly discovered in sewage. And then, of course, penicillin was looming on the horizon. So in the 1940s, penicillin was introduced at the start of World War II, and the Allied troops received penicillin, and our opponents were receiving phage, and we won. And penicillin was widely acknowledged as our secret weapon. And then we went straight into the golden age of antibiotics, and antibiotics were coming one after another, and antibiotics are actually pretty great. And so antibiotics were the treatment of the future, phage was the treatment of the past, and people in the West had no interest in phage. But in Russia and Poland, phage was used widely. Hundreds of thousands of people were treated with phage. Very little records, however, to document those results. And so now with our new century, we have the dry pipeline. We don't have antibiotics. It's been decades since we've had a new class. We have incredible epidemic of antibiotic resistance. And we're starting to see phage trials first in 2009 with multi-drug resistant Pseudomonas. And then a very watershed moment was in 2017 with Chip Schooley's group at University of San Diego with a successful personalization of IV phage therapy for a very sick man with Acinetobacter. So it's really been these case reports that have created tremendous amount of interest in our community. They really moved the needle in the United States to push people's interest in phage and really starting kind of at a ground roots level. And so right now there's about 50 phage trials in clin trials. Prosthetic joints are number one, but CF is kind of in that second tier. There's 7 trials listed, along with wounds, burns, and UTIs, and then a few other consequential infections. So with CF, we really have 2 targets. We have the gram-negatives, in particular Pseudomonas, but also Achromobacter and Burkholderia, and then Mycobacterium abscessus. And these are very different targets, so we have very different clinical designs here. The gram-negatives are an airway dweller. They form biofilms. They grow quickly in the lab. Abscessus can be intracellular or it can be a biofilm, and it grows slowly. The gram-negatives acquire phage resistance quickly, sometimes with just 1 dose. The abscessus does not appear to become resistant. So the treatment duration of gram-negatives are days to weeks, for abscessus a year or multiple years. And then the issue of neutralizing antibodies doesn't seem to be such an issue with gram-negatives, where it's very, very common with abscessus and other NTMs. So right now we have 2 lanes in the CF community. We have the compassionate use lane, and then we have the clinical trial lane. And we're getting a lot of information from both, and I just wanted to kind of cover some of these. There's been about 20 case reports, and some of them have been extremely informative. But more importantly, we have 2 really valuable consecutive case series that, again, we eliminate the bias of successful or interesting cases. And one of these was from Yale, and treating Pseudomonas, the other one from Pittsburgh treating Mycobacterium abscessus. And then we have 1 prospective observational trial, which is post-amp for the treatment of abscessus. And I wanted to just spend a second here with the consecutive case series. The one that looks at CF is the one that looks at NTM is from Graham Hatfield's group in Pittsburgh. He provided all the phage from his massive library of over 20,000 isolates. And 16 of the 20 were CF. Each row on this table is a patient. And I just wanted to emphasize the variability both in pre-transplant, post-transplant, peds, adults, 4 different species of mycobacteria, but especially the route, duration, and frequency of the dosing. And so when the clinicians were asked, in retrospect, did the patient respond, about 60% perceived an improvement with clinical response, and about the same perceived an improvement in microbiologic response. But there was a substantial minority that did not respond. And so the question was, could we replicate a successful case with a standardized protocol and improve the response and get a better idea of what to expect in terms of the response? So the case we picked was a case from Colorado that we were very familiar with. This was a 25-year-old man with abscessus, treatment refractory for many years. He was treated for about a year with phage therapy with significant reduction in percent positive cultures. And at the time of transplant, his explanted lungs were negative for abscessus. And so this became the treatment protocol that is the basis for the post-stamp trial. So the goal of post-stamp is to assemble just 10 individuals with treatment refractory abscesses and CF lung disease. They have to be on guideline therapy for abscesses and other comorbidities. The Hatfield lab will provide matching phages to approximately half of them. That's based on the rate of success that they've seen in the past. And then we'll facilitate regulatory approval and apply uniform clinical assessments of response and durability for a year of treatment and a year of follow-up, and then assess markers of response and treatment failure. So it's an observational study of people with CF who are receiving phage for compassionate use. And the second trial, or the second case series that I wanted to touch on, I think we'll hear more about this as well, but this is from John's group at Yale, that 9 individuals received treatment for refractory Pseudomonas, all CF, 1 to 3 phages that were nebulized over a course of 7 to 10 days, either once or twice daily. Most of them had at least some exposure to IV Pseudomonas antibiotics as well, and it was safe and well tolerated. And if we look at the burden of infection, every patient had a pretty sizable, some very dramatic drop in Pseudomonas CFU in their sputum. There was a few rebounds, but none to the point of the start. And there was a uniform significant improvement in FEV1. So at first this looks like a successful antibiotic trial, but it is much more nuanced because if we look at the rapid development of phage resistance, this is the table of every subject. Every row is a patient. Every filled dot is phage resistance. So if we look at if you look at the columns pre and post treatment, you see everyone developed resistance. And you're thinking that's no good, but it's not so bad actually, because there's a targeted trade-off. There's a fitness cost that the bacteria has to achieve to develop phage resistance. And so what the trade-off is, it loses other virulence. And so this is an example. 2 individuals got a phage that was directed against the MEX efflux pump. Those individuals had resensitization to antibiotics that they had previously been resistant to. Another example here is piocyanin that is linked to a phage, or there was a phage that was directed towards the type 4 pilus. It resulted in dramatic decrease in piocyanin, which is pro-inflammatory, but also it has that green color, so you can see just dramatically here it goes from the bacterial suspension goes from this bright green to yellow. So it's really, we think of like when we treat antibiotics, what we don't kill, they get stronger. But in this case, what we don't kill, they get weaker. And that has really important long-term consequence. So I wanted to touch then on the clinical trial space. Right now, there are 2 phase 1 trials that are upcoming. This is includes the acromophage trial at Pittsburgh, and then the taking advantage of phage therapy trial at San Diego, which is targeting people on treatment for pulmonary exacerbations for a variety of different CF pathogens, and we'll be receiving phage during that treatment. There's 3 completed phase 1b/2a trials. This includes the Armada Swarm study, an NIH-funded study, and then the first BioMX trial that is completed. And then the phase 2b space is the Sci-Fi trial from Yale that was abbreviated because of COVID, and then the current BioMX phase 2 trial for Pseudomonas. I just want to really finish by talking about the trial design considerations. The question we're asked most is IV versus neb. IV is reliable, but you do need IV access, and there is the question if phage really is going to get to the the airway in a very high concentration. Nebulized phage, you may get it in a higher concentration in the airways, and it's also linked to potentially less neutralizing antibodies. It may be more convenient for some patients, but it is probably less reliable, less standardized. There's the issue of phage could be in the room. That could be a problem, like during hospitalizations. And then there is the issue of device approval linked to the phage that can complicate these study designs. So we have examples both of IV and NEB, and I won't read them off, but the one I'm really excited about is the achromophage trial, which will compare both. This is Dr. Daria Van Tyne's study that's going to enroll in Pittsburgh starting in January, and it will compare IV inhaled and the combination of IV and inhaled. It's just 3 doses, but they're ascending doses. And so the email is phage@pitt.edu if you have a subject. The second thing I wanted to touch on was something that John mentioned, is just the selection of phage. The traditional personalized method is the plaque assay, where you would screen an isolate against a library and with a high degree of confidence. But that's not going to really work for commercialization. This is a really elegant paper that came out in Nature Communications from the BioMX group, and describes how they have gone about designing their cocktail. The personalized approach, you know you're going to get a lytic phage at the start of treatment, but there can be a huge delay in starting treatment. Banking and shipping isolates is not part of most labs' routine, and the isolate you send may not be representative of the infection, for these polyclonal infections, and it's also really not feasible for commercial applications. The fixed cocktail, that's very much like ordering an antibiotic from the pharmacy. It would be quicker, better standardized, and feasible for commercialization, but you do have this risk that an individual may not receive an active phage. So right now, we have a lot that's going on in the personalized space, and particularly the compassionate use group. We have at least one example of the fixed, and then we have kind of this hybrid where it's a fixed cocktail, but they're circling back and screening against the personalized isolates. So they're not personalizing the cocktail, but they are verifying that there is an active phage. And then I just wanted to finish with the idea of whether or not phage requires antibiotics. And so for the most part, phage works better with antibiotics. Sometimes there's synergy, sometimes it's additive, occasionally it's antagonistic. And so that you have this potential with antibiotics to get a greater therapeutic effect. And it also reflects treatment practice that, our very sickest patients are on antibiotics already, but it's much more challenging to determine the actual effect of the phage. It's hard to standardize the antibiotic combinations, and if there's going to be toxicity, it's probably from the antibiotics. So without antibiotics, you have a cleaner study design. You have the potential for regulatory approval as a non-inferiority study as opposed to a superiority study. But of course, it may not be feasible or ethical to stop antibiotics in an individual. And there is the question, if you don't include antibiotics and you don't reach your milestones, if the trial is unsuccessful because you didn't include antibiotics, you may regret it. So right now, quite a few of the trials have antibiotics. There's none that are like totally without antibiotics, but we are seeing this hybrid now that people are allowed to remain on inhaled antibiotics as standard of care. And then some of those individuals, their standard of care is not to be on inhaled antibiotics. So we're starting to see some data now on people who aren't on inhaled antibiotics, which will be very interesting. So I'm going to leave it there. So far, everything's safe. We're crushing the safety index, and that's what Deherel knew 106 years ago. We've learned a lot about phage resistance, neutralizing antibodies, and PK. We're definitely seeing promising trends toward decreased burden of infection and the potential for durable selection of less virulent infections. There's a lot of interesting, innovative study designs, but you really wonder, like, maybe it doesn't matter because the phage has been evolving for 3 billion years to find these bacteria, and if you just get out of its way, it's a good chance that a lot of these study designs will be successful. So I want to thank lots of people, the post-amp team at the TDN Coordinating Center here in Seattle, all the post-amp PIs and RCs at the NTM Consortium. And then I wanted to put in a plug for when you do these trials, it's really good to have translational and basic scientists involved that can look at these markers, both mechanisms of failure and mechanisms of success. So we have a large group, and we're looking to add more people to this slide. And then thanks to the Colorado Mycobacteria Phage Working Group and the NTM core at National Jewish that's funded by the CF Foundation. And then last and not least is my adult program at National Jewish for all their good care. And thank you very much.
Many thanks for an amazing presentation. I see some people at the microphone already, so go ahead.
This is yeah. My name is Paula Zamora from University of Kansas Medical Center. I was wondering, could you comment a little bit on the study design in terms of number of people enrolled? I mean, I know that some of the studies are only enrolling people with CF, and I'm assuming that maybe for certain like specific isolates. And what are you envisioning in terms of enrolling people to increase like the power of the studies?
Yeah, no, that's a great question. A lot of these studies are small. Like we saw the chromophage, they're projected to have 12. These are under the label as phase 1 or phase 1B. And so they're starting out pretty small. But I think, the hope is that there'll be a safety signal. We'd be able to calculate the power that would be needed to show, a phase 3 trial. I think right now, I don't think anyone really knows how big a phase 3 trial would have to be.
Paul Robinson, Brisbane, Australia. I was fortunate enough that I looked after one of the children that got Phage from Graham Hatfield's lab in that case series, and thankfully it was successful. My question is about ETI, and the question relates to the fact that when we started Phage and used it for a year, we used it IV, is that we also looked at other compassionate measures we could get, and invariably at that time ETI was one of them. And whilst I feel confident that it was the Phage that made the difference, I've often wondered about what role the ETI played in maybe setting the scene for us to have clearance eradication, and that child now kind of 4 years, 5 years on is still clear. So I just wonder in these studies, how do you adjust for ETI, or are you worried about whether some people are on ETI or what effect that might have to your overall success?
Yeah, no, I agree. And there has been some of these cases, it seemed like the ETI was very important in clearing the Mycobacterium abscessus. I think one of the ways I think about it is, we have to take advantage of everything we can. And with the abscessus, there is such a disparity between the Pseudomonas burden, which can be 10 to the 7th, 10 to the 8th, and the abscessus burden, which can be like 4 colonies. So you've got like 6, 7 log difference. And so anything you can do to reduce the Pseudomonas burden to allow the phage, to find the abscessus, I think we should take advantage of. And I think it'd be naive to say that ETI doesn't help, because we have microbiome data on our cases, and with ETI you see a decrease in Pseudomonas, and that's when sometimes you see the effect of the phage as well. So I don't think, these cases are completely due to ETI, but I think ETI probably does help. And, to the extent that you can thin the mucus, to the extent you can drop the Pseudomonas, then that's all for the best.
Hi, Jerry. Katie Heiser from National Jewish Health. I wanted to bring up one more aspect of trial design that you didn't mention, but I know you and I have talked about, which is how many doses do you give and how often do you give those doses? Because technically, as a lytic phage, it should self-propagate, unlike an antibiotic that gets used up. The phage, technically, if you were in a dish, it should just keep replicating. And particularly in light of, you talked about antibodies being made. The foundation has now made it clear that immune response to phage therapy is high on their list of interesting areas to study. Are we giving too much phage? Should we give it once a month? What do you think?
Yeah, no, and I think it just goes back to this idea of what are you targeting, and then John can talk about, with Pseudomonas, maybe a week is plenty. I think with mycobacteria, given the extremely slow dividing rate, that the experience has been that it takes much, much longer with mycobacterium. And I do worry that part of that is a reflection that we're not dosing high enough, and that's work by Miriam Bronstein's group that the phage is the mycobacteria is intercellular, and to really get intercellular, you need, it's kind of a dose response. And so, yeah, I'm a little frustrated by, how long it takes to treat. I don't know if we've seen much, in vivo evidence of phage replication. I don't know if our other speakers will talk about that as well.
All right, great, thank you. So our next speaker is Andrea Hahn, who's joining us from Children's National in DC. It's going to talk about the pediatric phage therapy landscape.
And just to say, there are a few. I don't know why this is. Oh, there we go. There are a few questions that are appearing up here. We'll ask them in the panel discussion at the end. So don't worry, we're not ignoring you.
All right. Well, I want to thank Jean and Isaac for the invitation to speak to you guys today. I'm tasked with talking about the pediatric version of phage and how we're targeting that specific population. And the short answer is, the data here is much smaller than what was presented thus far. I have no disclaimers and no disclosures to share with you today. So the agenda for my talk is going to be to summarize the compassionate use cases of bacteriophage in children with CF, to talk about personal experience with bacteriophage treatment in treating a patient with pediatric with CF, and then again to discuss ongoing clinical trials and areas for future research specifically related to pediatrics. So I'm really going to go over 3 good review articles that cover pediatric cases in what's in the literature so far. This is limited to cases that are, available and indexed in PubMed. So, we heard before about the long-standing history of phage use and how it's been around since the late 1800s. And so I'm not covering every single pediatric case. Again, and as we talked about before, there's probably a lot of cases where pediatrics have been treated with phage that have just not been reported in the literature. But again, just to kind of go over what is out there right now. So the first 2 cases, this is an article by Ben Chan, and the first 2 references are patients that were treated at the Eliava Institute. And so the Eliava Institute is in the country of Georgia. And so that is like a specific place where they've been using phage a lot, even during the use of antibiotics. And so have a long-standing history of kind of using phage in this population. And so the first case was a 5-year-old that was getting just standard CF care, but was continuing to grow both Pseudomonas aeruginosa and Staph aureus. And so this particular patient, they nebulized inhaled phage against both in a cocktail over 1 week, and they were able to report out reduced sputum density of the bacteria. as well as improved patient health and extended time to infection requiring antibiotic courses. And so this particular patient, they gave 3 rounds of treatment, and that is what they reported in their case report. The second patient here was also treated at the Eliava Institute. It was a 7-year-old female who, again, also was culturing Pseudomonas aeruginosa and Staph aureus commonly. And so they, again, gave a cocktail of inhaled phage against both of these bacteria. In this particular patient, they repeated the course 9 times every 4 to 6 weeks. And what they found is over that time, there was reduced sputum bacterial density, again, of Pseudomonas, and a 50% reduction of need for antibiotics in that 9-month period where they were redosing phage every 4 to 6 weeks. They didn't see any change in the imaging for that patient in the chest imaging, but they did note that the culture, at the end of the course, they were no longer culturing Pseudomonas or staph from their respiratory samples. The last case that I have highlighted at the bottom is a case of Mycobacterium abscessus. And so this was a 15-year-old patient who was infected with M. abscessus for about 8 months prior to a lung transplant. And then they continued to have significant problems with infection and ended up developing disseminated M. abscessus disease. And so they found an engineered phage, and this was mentioned in the talk before me, where they were able to treat this patient, was given IV a 3-phage cocktail, and they reported decreased size of the skin lesions that were present, improved lung and liver function as those were also infected, and improved chest imaging. And they noted that the M. abscessus was no longer culturable from either the blood or sputum samples, but it did continue to culture positive from skin lesions for an extended period of time. The next review article was by Nanda Ramchandar and John Bradley, and they did a nice review of pediatric compassionate use cases from like 2018 to 2025. And there were 5 cases that were specifically in people with cystic fibrosis. And so the first 4 are all cases of Achromobacter. And so as we heard before, this is a less common pathogen in people with CF, but one that's difficult to treat because it has a lot of intrinsic antibiotic resistance. And so their first patient was again treated at the Eliava Institute, received 2 phages in a cocktail that were again inhaled and also by mouth. And they reported that they gave the phages for like 20 days at a time and gave 4 courses over a period of 12 months. And then the patient had an improvement of their FEV1 from 54% up to 84% at the end of that treatment period. The second case was a 10-year-old that was treated in South Carolina that had pan-drug-resistant Achromobacter, and they were being treated with a combination of cefiderocol and meropenem vaporbactam in addition to the IV bacteriophage that they received, was hospitalized twice to receive this IV phage, and they reported improved pulmonary function. And at the end of the treatment course at 8 and 16 weeks after the IV phage, they were not able to isolate the Achromobacter from the sputum anymore. The third case was a 12-year-old that had a lung transplant. This patient had a history of growing a multi-drug-resistant Achromobacter and then had a pan-resistant strain, which led them to seek phage treatment. And so this was in France. They gave phage followed by imipenem and then reported decreased bacterial density of the Achromobacter that lasted for 16 months. And then after 18 months and then cultures at 26 months after treatment, they were not able to isolate the Achromobacter from the sputum. They also reported increased pulmonary function. And then the 11-year-old, the last patient with Achromobacter here, was treated at Rady Children's in San Diego. And this particular patient got 2 IV phages for their Achromobacter, also given with piperacillin tazobactam for a 2-week course. And as opposed to the other ones where I've just talked to you about sputum cultures and density in sputum cultures, they actually did metagenomic sequencing to understand how the phage was affecting the overall bacterial community. And they reported an 86% reduction in the relative proportion of Chromobacter that was detected 1 month after treatment. And this particular patient also had stable lung function during that time. And then the last patient was a 6-year-old with Pseudomonas that I'm going to discuss in a couple slides. This report was a case series that Dr. Nick also had shared by Rebecca Dedrick that showed 20 consecutive cases of NTM phage treatment. And of those, there were 6 that were pediatric-specific CF patients. And so these, again, were made at Pittsburgh, but distributed all across the United States, so UK, Italy, Australia, and the US as well. And as mentioned before, of the 20 total cases that they reported, there were 11 favorable, 5 inconclusive, and 4 with no response. And so some of these pediatric patients, the first 2 both passed away, but from complications outside of specifically related directly to the infection. The first patient died from transplant complications, and the second patient dying from adenovirus. The third pediatric case here, they did show improvement of clinical signs and symptoms after 7 months of prolonged IV therapy, but only had clearance of the rough strain of M. abscessus. The fourth patient had resolution of their infection after 1 year. The fifth patient had an avium, and they had improved lung function and clearance of cultures and is continuing to undergo treatment. And then the last patient with M. abscessus, they had improved chest scans, treating for over 1.5 years with IV, continuing to be smear positive and still undergoing treatment at the time of this publication. And so now I'm going to talk to you about a particular case that I was involved with treatment for. And so this was a 6-year-old Black female with CF. She had chronic lung failure, acute on chronic respiratory failure. So she was someone that had a pretty bad lung function kind of coming into this particular hospitalization, had grown multi-drug resistant Pseudomonas multiple times before, and now had a pan-resistant strain. And we tried multiple combinations of antibiotics during her hospitalization. So she was in the ICU, on BiPAP, pretty continuously. And despite 30 days of IV antibiotics, she still was not improving at all. The parents at the time were not really interested in pursuing a lung transplant. And so we were really kind of stuck at what else can we do for this particular patient? And there weren't any clinical trials at the time that were available. So this case happened back in 2021. And so we talked to the parents and had reached out to Yale University to see if they might be interested in trying to screen this patient to see if they would be eligible for phage. And so we were able to send a sputum sample, and they identified 3 bacteriophage that would potentially target this particular Pseudomonas that we had grown in culture. And so we reached out to the FDA and were able to get an emergency IND. And so because this patient, again, was in the ICU, this acute on chronic respiratory failure, it only took 2 days for the FDA to provide approval. And then we submitted that to the IRB, and 3 days later, we had an emergency approval by the IRB. And so, it was really helpful working with Yale and the FDA and the IRB that they all just recognized kind of the urgency of the case. And so we were able to do this quickly for the patient. And then after we got approval, we were able to administer the first phage. And so the 3 phages that Yale had identified, one of them had previously been tested because the phage are produced in endotoxin, so you need to know that it's safe to administer, while the other 2 hadn't been tested yet. And so we started with the first phage, gave it twice a day for 7 days, and then continued to give it with IV antibiotics for that patient. So with the first phage, she tolerated it without bronchospasm. So I had, told the FDA I would be present, and we observed that patient in the ICU for 30 minutes just to make sure that she was going to do okay. And then when she was able to do fine with that first dose, then we gave subsequent doses twice a day for that week. She was able to wean to high-flow nasal cannula during the day and was just doing BiPAP at night, and had decreased sputum production and increased energy. And so at that time, we were still kind of waiting for the second and the third phages to pass the sterility. And so once we did get approval for that to administer, the patient's family had had some discussions and decided that they did want to seek evaluation for transplant. And so as we're not a transplant center, we transferred her to Children's Hospital of Philadelphia. And while they were there, they had initially thought she would come back to us and just have the evaluation. But as she was there, they started to do, a lot more things like dental extractions and tracheostomy placement kind of in preparation for this lung transplant. And so because when you get an IND, it's like given to a specific physician, we couldn't just like transfer it over. And so I withdrew my IND, and then CHOP was going to pursue getting an IND to administer that second and third phage. But unfortunately, the patient passed away before that could be done or before a match could be found for a transplant. So the labs, I've just kind of glossed over those, but as you've probably been reading, they really didn't change too much. The only thing at the very end, like 3 months after the original phage, she did have a little bit of a bump in her AST, but again, whether that was related to phage or not is not clear. And so in the last kind of portion, I want to talk about ongoing clinical trials and areas for future research, again, specific to pediatric patients. So of all the clinical trials that were discussed in the talk before, only one is actually allowing enrollment of pediatric patients. So the post-stamp is a compassionate use for age greater than 6 years, again, enrolling by invitation. But all of the other clinical trials that were shown on that screen are not enrolling any pediatric patients. There are 2 studies in Australia that are enrolling pediatric patients. So the first is a single-arm, open-labeled safety and tolerability of a Pseudomonas phage. And then the second one is the standardized treatment and monitoring protocol to assess safety and tolerability of bacteriophage therapy in general. And both of those, again, are enrolling patients who are over 5 years of age. So, I understand that a lot of times due to safety concerns, we want to do clinical trials in adults, but there's really this long track record of safety. And so, I don't know that in and of itself should be excluding pediatric patients from these clinical trials. And I also just wanted to kind of point out a couple areas where it's really pediatrics is the place that you're going to need to answer these questions. And so one is like eradication. So with the initial Pseudomonas aeruginosa infection, there are, guidelines by the CF Foundation about how, we are going to go about trying to prevent chronic infection with Pseudomonas. And so there's a lot of an aggressive antibiotic treatment regimen that people use when children first culture positive. And so this may be an area where phage would actually be beneficial. And so, when you're designing the trial, you have to consider if you would just limit it to when standard of care fails. And so you've already done your inhaled tobramycin, you've already done, IV antibiotics, and they're still culturing positive. So maybe that would be a case where you'd add phages. Concurrently with standard of care, and so adding it in with the antibiotics that we're currently using with the inhaled tobramycin, or could you use it in place of inhaled tobramycin? And so, some of the other studies that aren't being discussed today, but trying to limit aminoglycoside use for treatment of Pseudomonas because of the ototoxicity and other complications, that if you could use phage in place of inhaled tobramycin, that might be something that would prevent long-term problems for our patients. Another area that would be unique to pediatrics is prophylaxis. So I know in the US we don't typically prophylax Staph aureus, but that may be something, instead of using antibiotics, phage could be used. And so I think that one of the take homes for you is just that pediatric patients, like there's an opportunity for unique trial design that could address questions that are specific to this population. But also in general, I think they have a lot that they could benefit from phage just as much as adults, and we don't want to leave them behind. So key takeaways, so bacteriophage for several CF pathogens have been used in pediatric patients on a compassionate use basis. And so Chromobacter, Mycobacterium, Pseudomonas aeruginosa, and Staph aureus. But the ongoing clinical trials to study phage, unfortunately, are very limited to include pediatric patients. And I think pediatric people with CF, they have unique scenarios where bacteriophage may be beneficial compared to adults that we're studying right now. So I thank you for your attention. Happy to take any questions.
Amazing. Thank you so much for that presentation. I see one person at the microphone already.
Yeah. Hi. So you mentioned a few times the Liava Institute. I'm wondering from a kind of like physician perspective or a phage therapy center in the US, I know that the Liava Institute and the phage therapy unit in Poland, they have their own phage libraries and, they have been treating kids, as you mentioned, for many, many years. So would you comment in maybe collaborations with the centers in Europe that they have libraries and more experience, and also commenting in maybe the idea of a centralized phage library instead of now that it seems that it's individual institutions that are creating their own library. You know what I mean? Yeah. Thank you.
Yeah, so I unfortunately would have no power to make that wonderful idea come into fruition. But, I do think you bring up a big point that there's a lot of institutes outside the United States that have much more experience and are more comfortable with using phage. I think I mentioned the 2 clinical trials in Australia where they're allowing enrollment of pediatric patients. And I think maybe we could take a lesson from them in trying to involve more pediatric patients. But as far as, phage libraries, and I think it's academic centers are definitely, willing to work with each other to try to make phage accessible, but with industry, trying to figure out how to get over the hurdle of making this into a product that they can deliver, and all the regulatory hurdles. I think there's a lot of different pieces that maybe would make it hard to have just this one phage library that everybody would have access to.
Fantastic. Thank you so much.
Thanks.
Great. So it's my enormous pleasure to announce the next speaker, Dr. Katie Wetzel from Georgia State University.
Hello, everyone, and thank you so much to the organizers for inviting me to speak today. As was said, my name is Katie Wetzel, and I'm an assistant professor at Georgia State University, but I did my postdoctoral fellowship in Graham Hatfull's lab. And so I'll be changing gears a little bit from the talks we've heard so far today, whereas I'm really a basic phage biologist. And so I'll be telling you about work done by myself and others to try to better understand phages that infect NTMs and what we can do to make the phages that we have better and really just improve on phage therapy for these infections as a whole. As we've heard, a few times today, especially if you were in some of the earlier talks, NTMs, or non-tuberculous mycobacteria, are a problem for people with cystic fibrosis. And in the most recent CF Foundation patient registry, about 12% of people had a positive NTM culture. And these NTMs, of which there are hundreds of species, are mainly for what we're concerned about, M. abscessus and M. avium. These infections are very difficult to treat, and the antibiotics that are used are often really poorly tolerated by patients. And so alternate therapies are very much needed, and bacteriophages are such a therapy that have shown promise. I want to start by talking about the phages that we are using for therapy. So as some of you may know, what's sort of the common thing is when you have a bug for which you want to target with phage therapy. If you don't have any phages for it, you go out in the environment. You get some wastewater, you get some hospital runoff, and look for phages there. And for M. abscessus, which is the NTM that I'll be talking about most today, and really the other NTMs, that hasn't been as fruitful of a strategy as it has been for some other bugs. And so what really ended up working out, you could say, for phage therapy against NTMs was using a collection of phages against a model organism, M. smegmatis. So the lab of Graham Hatfull at the University of Pittsburgh and the Howard Hughes Medical Institute put on an undergraduate research program called C-phages. And so what this program does is undergrad students throughout the US and in institutions throughout the world really go out into the environment, get some dirt, and get some water and use that to isolate phages against this very safe, rapidly growing model organism, M. smegmatis. And so because of that, the Hatfull lab has thousands of phages that infect mycobacteria. What those phages look like are shown in this EM. We've seen a few pictures so far, but they've got a double-stranded DNA genome that is in this capsid here. Most of our phages have this long, non-contractile tail. And then at the very base of the tail, what we have is a tail tip with a bunch of different proteins that interact with the bacterial cell and presumably confer some specificity. And among our mycobacteriophages, as we call them, there's lots of different tail tips, lots of different proteins that exist among these thousands of phages. As we've heard a little bit about, some of the phages that were isolated on M. smegmatis immediately are excellent at just infecting and killing the target cell, and that's the only thing they do. Other phages might sometimes integrate their genome into the host cell, hang out there for a while before they replicate, and as was discussed, that's not what we want for phages used in therapy. So some of the phages that have been isolated on M. smegmatis, if we want to use them for therapy, we need to engineer them to only be lytic, only kill the cell every time they infect him. So we've heard a bit about phage therapy to treat NTM infections in our first couple talks, but I'll run us through it again, but this time provide a little bit more context and take a step back. So the Hatfull lab historically, did not study phage therapy. Graham Hatfull is a basic phage biologist, but back in 2017, he was contacted from a care team of a pediatric cystic fibrosis patient in the UK who had a disseminated M. abscessus infection, and she really was doing quite poorly. So, the care team knew about the Hatfull Lab's collection of M. smegmatis phages and asked, hey, would you be willing to screen your phages against our patient isolate to see if you have anything that might infect and kill that strain? So the strain was sent to Pittsburgh, and my colleagues, Carlos Guerrero and Becca Dedrick, who's here in the audience, went through the challenging task of screening this isolate with hundreds of phages, looking everywhere they could to find a phage that might infect and kill this strain, and they ended up finding about 5 phages that were mostly M. smegmatis phages, again, that were able to infect and kill this strain. And so ultimately, a cocktail of 3 phages was given to this patient, and what you see on the right-hand side are her skin nodules that were infected with M. abscessus pre and post-therapy. So after several months of phage therapy, she was able to return to school, return to her life, and that really launched, the NTM phage therapy program out of Pittsburgh that we have today. So again, to then put that into context, we've talked a little bit about the case series that described 20 patients who were treated with phage therapy. but those 20 patients came from 200 clinical isolates of M. abscessus and other NTMs that were screened for phage susceptibility. And of those 200 isolates that were screened, phages were found for just 55 of those. And then it was 20 of those patients that were ultimately treated with phages. And as we've already heard about today, just over half of them improved, whether it was by clinical measure or by decreased bacterial burden. Importantly, again, as we just heard very well described in the last talk, phages have been very well tolerated, and this providing of phages for compassionate use treatment is something that the Hatfull Lab continues to do. They are at about 48 patients that have been treated with phages. This includes participants in the post-stamp study that we heard about, and we're still learning about a lot of these patients, how it's going to go for them. As I mentioned in the introduction, where someone like me really comes into play is I'm really interested in better understanding these phages. How do they recognize their host cells? How do they infect them? What do they need from the cell to replicate? And myself and scientists like me would argue that if we can better understand just this basic phage biology, we'll be able to leverage that into improve phage therapy and provide it to more patients. In doing all of these screenings of different patient isolates for phage susceptibility and the downstream treatment, a bunch of different challenges have emerged. And so I'll go through some of those now. The first challenge is that phage susceptibility varies really unpredictably among NTM clinical strains. So what you can see here on the left-hand side, these are 3 different phages, Muddy, BPSH-RM10, and Zoe J Delta 45. And these are the 3 phages that were used in the cocktail to treat that very first pediatric CF patient. And so you can see that when we take these phages and serially dilute them onto lawns of bacteria, we get these nice zones of killing, these nice plaques showing us that our phages can effectively kill our strains. And we see that here with our model organism M. smegmatis, as well as that first patient isolate GD01. However, when we spot these phages on M. abscessus strain GD10, we see that only this first phage, Muddy, is really able to infect and kill it. And GD10 is a very close relative of GD01. And we're still very much in the early stages of understanding what these differences are. The second challenge is that we know very little about how M. abscessus and other NTMs can evolve resistance or even co-resistance to our mycobacteria phages. And we've talked a little bit about this already in this session, but when we are treating with phages, a common strategy is to treat with more than one. And that was certainly done with the very first phage therapy cocktail that was given. She received, as I mentioned, these 3 phages, and these 3 phages were chosen because they are all very genetically different from each other. And so the rationale was it would be very unlikely for the bacteria to evolve, resistance to 3 phages, presumably by 3 different mechanisms. An important point that was made by Jerry already is that for NTM phages and NTM-based phage therapy, we're sort of in a good but unique position in that we generally have not seen phage resistance emerge in a patient post-therapy. But we can do it in the lab, and we want to make sure we certainly get ahead of this before it potentially becomes a problem. I am very interested in addressing these 2 challenges, and I've been doing that by asking the simple question, how do phages interact with the mycobacteria cells they infect? And so to do that, in my last couple of years as a postdoc in Graham's lab, as well as in my own research lab now at Georgia State, I've been doing genetic screens. So for these screens, what I do is I take an M. abscessus, at this point that's the only NTM I've been working with. I take an M. abscessus strain of interest, make a mutagenesis library, take this mutagenesis library and put it on a plate that's seeded with phage. And then what happens is most of the cells die because they're susceptible to phage. But some of my mutants in my library are newly resistant to phage because they have an insertion in a gene that the phage needs for replication. And so you can see I can isolate these phage-resistant mutants and confirm that they are in fact phage-resistant, and then figure out where my mutation is, generally by PCR. So I did this mutagenesis strain with, or our mutagenesis screen rather, with one of our workhorse phages, BPS-HRM10, and I was able to characterize 18 phage-resistant strains, and for all of them, they had an insertion in one of the genes of this 5-gene cassette. And what this cassette is, it's for the synthesis and transport of a molecule called trehalose polyphleates, or TPPs. And so these TPPs are just these huge glycolipids that sit on the outer surface of the mycobacteria cell. And yeah, all these gene products helped export it and modify it. We don't know a lot about what TPPs do yet. That's an area of future interest. What was really surprising to me is I did the exact same screen with our another big therapeutic phage called Muddy, which again is very genetically different from BPS. And so I was really surprised when all of my mutants that were resistant to Muddy also had insertions in this synthesis pathway for trehalose polyphleates. We then went on to characterize this further, and this was a project that I did in collaboration with Laurent Kramer's group over at the University of Montpellier. And so what you can see here is that when we do our dilutions of phages on our TPP deficient mutant, we don't really see many plaques for BPS-HRM10 or Muddy. Importantly, we do still see many plaques for, equal to wild type for phage ZOE-J delta 45. So it's not a pan requirement for TPPs, specific phages require these. And so I'm summarizing a lot of data with this cartoon here. But what we found is that, these genetic mutants do lack TPPs on the surface of the cell. And we found that when TPPs are missing, the phages don't bind well. One of the things that we did in, fleshing out this mechanism is isolating mutants of phage BPS-HRM10 that were able to infect the TPP deficient mutant. And that we can do pretty easily because generally with our really, high titer preparations of phage, there's, random mutants that happen just in the generation of, billions and billions of phage particles. And we can isolate these mutants. And what I found is that every mutant that was able to infect the TPP deficient strain had a single amino acid substitution in the tail spike of the phage. So that's one of the proteins at the very base of that long tail that I showed you all earlier. And so these phages with these amino acid substitutions are now able to bind to the cells even though TPPs are not present. And one of these phages, which is called PHKSW1, is now used in therapy as a replacement for the original BPS-HRM10. So to summarize what I've told you so far, what I've found in the screen that I just showed you and screens that, unfortunately I don't have the time to show you the data from today, we're finding that phages that are really genetically different can rely on the same bacterial factor for efficient infection. And so it's really important that we investigate these mechanisms because we're finding, at least, in the lab, mycobacteria are evolving co-resistance to phages in really surprising ways. And we want to be able to understand these pathways so we can ultimately design good cocktails that are actually doing what we think they're doing and covering a breadth of possible resistance mechanisms. I showed you too that, in investigating these resistance mechanisms, we're able to isolate really new and better phages that circumvent resistance mechanisms and sometimes even kill better too in our in vitro assays. In identifying these different factors that are required for phage infection, something that my current lab is really interested in is better understanding how the presence and absence of these features is dictating what we observe in terms of the variation in phage susceptibility. So some of this could be that, certain strains are simply, mutants and they lack these factors that the phages require, but we also need to start thinking about how expression of these different factors might change in a CF lung, for example. And a really interesting question that maybe some of you in the audience have been thinking about throughout this session is, well, how are we not getting phage resistance to in our NTMs? Why is that not emerging in a patient? And so it's really tempting just to think about the fact that maybe these phage-resistant mutants are really, really unfit. And some work from Laurent Kramer's group that came out last year did show that M. abscessus that lack TPPs really don't survive very well inside macrophages. And so that's just sort of the tip of the iceberg in terms of understanding how phage resistance might result in really unfit NTMs. And so with the last couple minutes that I have here, I wanted to touch on some of the research that's being done by others to try to make phage therapy better, but again, from a sort of a basic standpoint. So the third challenge I want to discuss is that we have a limited number of phages. There's really only about 9 different phages that get considered for treatment of NTM infections. And something I sort of skipped talking about is that those of you who study M. abscessus know that it comes in a smooth and a rough morphotype, dependent on expression of what's called glycopeptidolipids. And we don't really have many smooth strains that are susceptible to phages, which is a challenge. And so our work in expanding phages falls into a few categories. One is that some of the mutants that I've already talked about, like phage PHKSW1, just sort of the mechanism by which it circumvents resistance kind of seems to just make it have a broader host range in general. So some of these mutants that we've isolated over time are just better at killing more strains in general. So we leverage that in our collection. Another thing that's pretty neat are these phages that we call LipSips. So that stands for lytically propagated, spontaneously induced prophages. So you can see why we call them LipSips. And so what these are is our M. abscessus strains often have, many different prophages in them. So those are integrated phage genomes because, the phage is lying dormant and thinking of a good time to replicate, sitting and waiting. And so what we can do is we can take some of our M. abscessus strains that have these prophages, sort of combine them with M. abscessus strains that are pretty susceptible to phage, and just sort of mix them together and see if anything comes out. And so that's what's shown by this cartoon here. And sometimes we do get spontaneous release of new phages. And so then we need to modify those phages a little bit. But then there is one phage in the phage therapy arsenal that is its story. It was originally a prophage in M. abscessus, and it was induced, modified, and now we have it as a tool. Lastly, as Jerry talked about, one of the challenges with phage therapy against M. abscessus and other NTMs is there certainly are times where neutralizing antibodies that are raised against the phages can be a problem. And this was, a case study where this was really clear, came out in 2021. And so I'd just like to point out the efforts of Krista Freeman's lab, and she's at Case Western Reserve University, and Raphael Park, who's at Scripps Research Institute. And so they both use advanced microscopy techniques to really look at the phage particles. And so that's what you can see here. This is a cryo-EM of a whole mycobacteriophage, and this is the first one for which we have an image at such high resolution. And so what we can do with this information is now we can see, and using mass spec as well, okay, what proteins are actually in the phage particle, what is exposed, what might we be able to chop off now that we see the structure. And so those two scientists are working hard to engineer phages that are less immunogenic and also best at infecting. So with that, I'll finish up. I'd like to thank my fledgling lab at Georgia State University, the Hapful Lab, for which there are many people, pretty much everyone has touched phage therapy at this point, our collaborators, the CF Foundation for my postdoc funding, and thanks again, and I'm happy to take any questions.
That was fantastic. As people come up, just one question occurred to me. So if the TPP story is fantastic, but is there any cost for the TPP independent phage therapy in a similar question? And it also had me considering, if you're removing more and more of this rough strain, are we also seeing increased cost or virulence of an increased smooth population?
Mm-hmm. Those are really great questions. So first, about the TPP independent phage, I think it asks, or it addresses sort of the broader question that we have these really, we use multiple in vitro assays to determine which phages are the best at killing. And so the TPP independent phages in our assays are very good at killing. But does that translate to actual better therapeutic killing? And we don't know that yet. And that's something I'm very interested in pursuing. And then your second question about, do we see, sort of kill the rough, see the smooth come back? I haven't heard of that happening yet, but it's, certainly something to be aware of.
Yeah.
Mm-hmm.
Wonderful. Sorry, yes, on the right, we'll go.
All right, Liz Bergner from Children's Hospital Los Angeles. I was really struck by your kind of anecdote about the first 3 phages to treat that young child with the skin lesions. They were isolated from M. smegmatis. I mean, I think of phages being so specific, right? It's not even broad enough to kill all of one species, but then to have it being cross-species. Can you speak to that, or are those 3 phages still pretty broadly effective, or?
Mm-hmm.
Yeah.
Yeah, so what I can say is, right, we have, thousands of phages that have, and about 2,600 at this point have been sequenced, and we, sort of put them all into groups, and the groups are like about 35%, shared gene content. And we have about 40 of those groups, and there's about like 4 or 5 of them. So 4 or 5 genetic groups of mycobacteriophages that seem to have expanded host range. So they will infect SMEG, some of them will infect TB, some of them will infect abscessus. So there's sort of a subset that seems to be a little bit more promiscuous, which is really different from exactly like you're saying, what you see for a lot of other phage bacteria combinations.
Yeah, that's really cool. Thanks.
You're welcome.
Hi, Gail Stanley from Yale University. Thank you for a wonderful talk. I wanted to ask for the NTM phages that you used in patients. Have you characterized their targets, and how often in the cocktail did you find that they had the same target?
Yeah, so that's something that we still really don't know, like in terms of like receptors and things like that. Yeah, we really don't know much about their receptors or what they target. So the TPPs is really the most we know. That some phages infect better when TPPs are present and other phages don't. For the other phages, we don't know. I've been doing more screens to try to find out. What pops up more often than not, honestly, at this point, is not necessarily stuff on the surface of the cell. So I've mostly been doing like, things that screen for non-essential genes. So there's a good chance that some of our phage receptors are just really essential things, and it'll take some work to find.
So how often in your cocktail were they all targeting or the TTPs then?
So rarely, but I think occasionally. That's a good question. I have to go back and look, because like BPS and Muddy are 2 of the phages that people get the most. And so there's probably instances where
So just thinking of the arguments between cocktail and single phage, and sort of like preventing the neutralizing antibodies, could there be to highlight how important it is to really characterize the phages that we're using, even though they're genetically distinct, to make sure that they have different targets when we're really trying to design a cocktail, or if we think that maybe a single phage would be just as good.
Yes, and a lot of the time, at least, that Zoe J phage that doesn't rely on TPPs was there, and so that helps circumvent that, at least in terms of resistance.
We're actually going to have to hold off on the additional questions just in the interest of time. So thank you.
Great. So it's my pleasure to introduce Dallas Mould from Yale.
Thank you so much. I just wanted to say I was part of the C-Phages project at UCLA, and it's a great way to find phages, but also a way to promote interest and persistence in science, and that's part of what led me to pursue a PhD. But I'm also very grateful to be part of the Turner Lab in the Ecology and Evolutionary Biology Department and a member of the Center for Phage Biology and Therapy at Yale, where I'm exposed to not only basic science, but translational and clinical aspects of phage biology. And a lot of what I'm going to talk about today is work done by my colleagues Ben Chan and Katie Courtwright, where I'm going to talk about some of our clinical cases, but also some in vitro work investigating whether or not single sequential phage therapy approaches and how they compare to cocktails, as kind of the questions alluded to. I have no disclosures to announce, but what I love about the NACFC is the diverse perspectives and the unified mission. And so I think it's important that I tell you kind of my background, which is in molecular microbiology. And so I'm a trained molecular microbiologist, and often I think about the clinic, but I'm really focused in these controlled in vitro settings and thinking about how much are these predictive and what can they tell us and predict about what might happen in the clinic. And so my conceptual framework, again, is quite simple. I think about the target, in this case, multi-drug resistant and pan-drug resistant bacterial pathogens. And most often, this is Pseudomonas aeruginosa in my case. And I think about the context. This is the physical location, the airway, but also the nutritional profile of the other microbes that are present. And this intersection is important to think about when we're applying agents like phage. And so phage is the rage, but for a long time, we've used antibiotics. And I think we have a lot to learn from our past and our history with this antibiotic focus in thinking forward about how we're going to move phage therapy into the future. And so we know that antibiotics can dampen infection, but as we've heard, resistance, right? So they can become resistant to antibiotics that they have not been previously exposed. And you can see this with phage as well, because resistance can also be mobilized for antibiotics as well as for phage. And ultimately, resistance is outpacing discovery for antibiotics. And I think this may be different for phage, but I think it's important that we think about, just like we do with antibiotics, how we can be good stewards of phage as well into the future. And so, as we heard earlier, phage therapy is not new. About 100 years ago, when phages were discovered, they were almost immediately used in insects, locusts actually, and then in this 12-year-old boy. And as you heard, their test of safety was just, let's drink it. And if we don't get sick, let's give it to the boy. And fortunately, he cleared this infection. But we've come a long way in thinking about safety, right? And so I'm showing you some of the cases here at Yale, not here, but where I'm from here, Yale, with aortic valve infection, but also in the case of cystic fibrosis. And it's really been nice as a basic scientist to witness kind of the translational aspects and where the significant impact this can have on the community. And so looking broadly across these larger scale studies where they've treated a variety of different pathogens, so different targets, in a variety of different ways, we see that there is still a margin for improvement. And really, we need to be thinking about when and where will phage therapy be most effective and what approaches should we be utilizing. And from my perspective, I like to think about those things that we can easily measure in people, but also those things that can be easily manipulated or tweaked without applying additional burden to these individuals that are receiving therapy. And so what do I mean by effective? And so first off, it's quite obvious, we want to get rid of the bacteria, right? And ultimately, this leads to people feeling better. And this last one, this trade-offs, we kind of heard earlier, but this perhaps is not as obvious for those that don't think about evolution. But we know that just like antibiotics, especially for gram-negatives, that resistance to phage is evident. And so through in vitro experimentation, we try to look at these phage-resistant isolates across a variety of backgrounds and see what their phenotypes are. And we try to choose our phages very carefully such that they have clinically beneficial trade-offs and these consequences to their fitness that might impact the clinical efficacy. And so just to show you this in action, you've seen some of this plot earlier, but this is work by Ben Chan, as well as in collaboration with the Cough Lab. And so we have 9 people with CF that received nebulized phage therapy, either cocktails of 2 or 3 phages or a single phage. And this was repeated daily or twice a day, depending on whether they were an inpatient or an outpatient. for 7 to 10 days. And you can see that there is a dramatic and significant drop in the bacterial burden after 14 days. And this kind of stabilizes off in some cases, but largely lower than what they started with. So we see that kind of first metric of efficacy, this drop in bacterial burden. And ultimately, this led to improvements in airway function. And so even if some of these differences might seem small, this has an impact for these individuals. But when we see bacteria dying at this kind of significant difference, we need to think about resistance. And I think this resistance is not surprising, and it's expected, and we need to think about it moving forward. And this is because bacterial populations are quite large, right? And as a result of growing and replication, this is an air-prone process. And so this creates heterogeneity of alleles. And these alleles are subject to selective pressures, and phage are a very strong selective pressure. And quite simply, those alleles that are susceptible to the phage or whatever we're applying, they're going to die, they're going to drop in frequency, and those that are favorable will rise. And this resistant isolates, they can either be through receptor, as we heard, through intracellular defense systems. But we try to take advantage of that resistant phenotypes in thinking about these trade-offs. And so there's this concept of the Darwinian demon, where you would become physically optimal across all conditions. But fortunately, this is not a reality. This is more of a conceptual kind of thought experiment. And so as you become more fit in one environment, there are evolutionary constraints. So this is not unique to Pseudomonas aeruginosa. This is not unique to E. coli. This is a common of all living things. And so as a result of this constraint, they have trade-offs in various phenotypes, and we try to look at that probabilistically using multiple lines and multiple different strains to look at drop-offs in virulence properties or antibiotic susceptibility. And so I'm showing you one individual's antibiotic susceptibility screen, and you can see at the start, they were resistant to this entire panel of antibiotics. But after the delivery of phage, we find that in that shift of color, that there are intermediate and susceptible isolates. So we are renewing and providing a new option even when phage resistance emerges, and we expect it to. And so with essentially a century of safety, maybe we value less just drinking the phage and you don't get sick. But we have some signs of efficacy as well, and it's time that we think about not only these discovery aspects, but also the deployment of these phage. And how are we going to prolong the utility of phage? And so phages are very abundant. We heard this number as well. There are 10 to the 31 estimated on Earth. Just to give you a little bit of perspective, if we were to line phage, remember these are very tiny, smaller than bacteria, just end to end, they would orbit the sun and the Earth 1 trillion times. Okay, so there is an extraordinary amount of phage in the environment, but it does take time to pull them out. And for some microbes, this might require additional effort. And so we have libraries, and I think that there is a lot of promise in the potential, unlike antibiotics, which are a little bit more limited. But we need to think about how we prolong their use in the delivery. And one thing that we've learned from antibiotics are some potential approaches like cycling of antibiotics or applying them in combinations, as we heard a little bit about. And what are the differences here? And so one thing I'm going to talk about today is this choice of whether we use one phage, a single sequential, so one phage and then another, or these cocktail approaches, and does it matter for what happens to the bacteria? And so Katie, Dr. Courtwright, chose to test this using experimental evolution. We essentially allow the bacteria and the phage inoculate together and look at what changes. And so she used 3 independent lines across 5 different strains. I'm going to show you the data from MPA01, but she also did this with 4 CF clinical isolates. And though some of the details differ, kind of largely 3 of the 4 CF clinical isolates show the kind of same big picture results I'm going to report here today. And so every day for the monophage treatment, she exposed them to one phage again and again and again for the course of 3 days, or a cocktail of phages targeting different parts of the bacterial surface, or in sequence. And what you can see, perhaps not surprisingly, is that the monophage, there's some decline, but not really over the course as evolution resistance emerges, but the cocktail is a significant decline. But what I'd like to show you is that sequential, you can have the same drop in bacterial burden. You can see our best sequence on the far right. But we have to, again, think about resistance. And here I'm showing you a kind of graphical schematic of the best case results, where we're looking at the susceptibility to at least one of the 3 initial phages. And what you can see is that when you deliver a cocktail, quite quickly, they become resistant to all 3 phages in these experiments. Whereas the delivery of a single phage, you get resistance to that one phage, but they remain susceptible to the other 2. And in sequential, we can extend the duration of our library, and of this case, just a set of 3, that they do develop resistance to all 3 by day 3, but this period is extended. And not only that, but we have to think about the mechanisms of resistance, right? And so here I'm showing you some of the numbers that go into some of these statistics, looking at the control. So just as you passage bacteria in the absence of phage, there are going to be mutations that happen. You can see the number there in that first gray bar. And then kind of following the monophage therapies. And then you see this bar kind of skyrocketing above the rest, right? This is our cocktail. So we see a lot more genes changing when you deliver this 3-phage approach. And this is less so with the sequential and even less in the control. And this is important to think about, not only from the trade-off perspective, because if we have a lot more genes changing, we might not be getting that intended clinical trade-off that we're looking for. Through our intentional approaches. But also, you have to remember about that heterogeneity, right? So if we have a pool now with many different alleles, they're primed that if we're not able to eradicate, they're primed to respond to additional treatment. And so just as you saw before, when we treat them with another therapy, maybe now antibiotics, they'll drop those that are deleterious, but again, we're going to have a rise. So this is something we have to think about moving forward, not only to prolong their use of phage in our libraries that may be more limited for some organisms, but I hope that some of the data I showed today, you can see how single sequential could have this similar significant drop in bacterial burden while extending the utility and making sure, or at least trying to push towards those clinical trade-offs that we're hoping for and leading to less off-target phenotypic consequences that may enable them to respond to other treatments. And just for the last tidbit, I'm going to talk about some of work that I've done in thinking a little bit about another aspect that we've learned from antibiotics. And so with beta-lactams, bacteria can make beta-lactamases, right, that degrade and get rid of these antibiotics. And so we have delivered beta-lactamase inhibitors with these antibiotics, right, to extend and expand their efficacy. And so we heard a little bit about phage defense systems, and kind of my thought is thinking about what adjuvants we might deliver to prime our bacteria to be killed by these phages. And so again, I'm a molecular microbiologist, and I think a lot about these controlled experimental systems. And one of these common phrases, if you spend an hour in the library, you can save years in the laboratory, which is very true. We should all read. And so almost as early as phage were found, calcium has been described for a role with phages. And in fact, this has become so ingrained that a lot of protocols for how to amplify phage suggest including calcium. And so I've screened all of our Pseudomonas phages that are commonly tested clinically, and about 42% of them actually require calcium. And in the literature, this extends to a variety of different bacterial genera. So this is beyond Pseudomonas aeruginosa. But what has been assumed historically, and I don't have time to show you all the data in support of this, but I'm happy to answer questions about it, is that it's thought that calcium mediates that initial step for phage absorption. But what I found by, again, looking at all of that phage collection, that only 31% of our calcium-requiring phages show evidence of absorption defects. And in fact, the other 70%, there seems to be an intracellular mechanism at play that happens post-genomic entry. And so there's this defense system that seems to be activated by calcium restriction. But does this matter in vivo, right? And some people might be surprised to think that it does, because when you look at total calcium levels in CF, it's actually higher than non-CF comparators, so about 1 to 3 millimolar compared to about 0.7 to 1.5. But what I'll argue is that the accessibility to the calcium likely varies, just like similar stories we've heard with iron as well, if you're familiar, and that there are a variety of proteins and sugars, carboxylates, that are secreted by other microbes and host cells that can sequester that free ionized form and perhaps impact phage performance. And so to get at that, I took sputum from people with CF and I homogenized it. And I was able to split it up into different tubes because I love my replicates, right? So I was able to have replicates of my sputum even from small sample volumes. And I spiked in a tagged susceptible bacterial host with a matching phage. And so in this way, I'm kind of circumventing delivery and whether or not there is a susceptible target in there. And I'm treating this as a media, essentially. And so for the sputa that I'm going to show you, these are all sputa that did not support replication of this calcium-dependent phage. And so I added water or calcium. I'm going to show you some of that replication data. And so you can see in the water condition, they kind of stay along the dotted line. This is for a very short period of time. This is the amount of time it takes for the phage to undergo 1 round of the lytic cycle in vitro from optimal conditions. And so you can see that there's a little bit of a drop off in the water. This is likely due to adsorption. And then you can see in the addition of calcium in sputum from a variety of different people, each stop being a different person, that calcium can support or promote replication of phage. And since this is a short period, I imagine that over longer extensions, we might see a greater amplification. And so ultimately, in summarizing, I think these are all approaches in how to think about fine-tuning phage therapy. We have phage are very abundant, and something I didn't mention, I forgot to mention, there was a talk about, that phages also co-evolve, right? So we can train our phages to extend their efficacy. But I think it's important that we are good stewards and think about our libraries and the very great potential for resistance. And I think that using single sequential approaches, we might be able to reach similar efficacy in terms of drop in bacterial burden. And this is something we should continue to explore. But that in doing so, we might also be able to slow the development of resistance to phages and also fine-tune what happens to the bacterial population afterwards. And lastly, calcium may be a good phage adjuvant, not only for limiting these phage defense responses, but also for those that are limited for adsorption. And so it seems like calcium may be broadly useful and might be something that is incorporated when delivering phages to people. So with that, I just want to thank the lab, and again, Katie Courtwright for the single sequential data, and Ben Chan, who was the lead on the clinical phage therapy cases. Thank you.
And Dallas, you get extra kudos because you were able to make this and give this wonderful presentation when one of our colleagues couldn't. So thank you also for that. Any questions from the audience? I can't. Okay, and so one thing that occurred to me is as we think about the discussions in the conference about the complicated sputum communities, is there a role for the anaerobes or other bacteria to modulate some of that calcium availability and to interact here in terms of kind of a multimodal interaction that we may have to anticipate when we're thinking about phage therapy?
Yeah, so I think this will expand as I evaluate more and more sputum samples. But what I've seen so far from the ones that I evaluated, these are all staph rich. And what I found in kind of co-culture-based assays by looking at the staph from the sputum samples, that staph can inhibit plaque formation of Pseudomonas targeting phages in a calcium-dependent way. But this depends on the staph, but maybe staph is playing some role in that. But I'm kind of working towards looking at degrading sputa in different ways and looking at, our protein or, adding staph targeting antibiotics. And then in that way, I can do these spike-in experiments and investigate what is a strong contributor and across how many sputa. And that's kind of the nice bit of having this individualized rather than a pooled sputum approach.
Fantastic. Go ahead, please.
Thanks so much for the great talk. Jonathan Raymond, I'm a respirologist from the pediatric clinic in Vancouver. So I'm not a phage biologist, so forgive me if it's an ignorant question. Do you have any evidence of coevolution of the phage to chase down these resistance mechanisms that crop up? Because I mean, it's a was it living or not? I mean, sure, but it's a biological entity. So does the phage you start with, is it the same as the phage you end with? Is it chasing these resistance mechanisms?
Yeah, so in vitro, there are a lot of examples of coevolution experiments where the phage do change. And so as maybe the receptor changes, often we find some tail fiber mutation that enables them to either use a slightly different receptor or overcome that mutational difference. So definitely that's something that happens. Whether this happens in vivo, I'm not sure. We can try to isolate phage post and look at that.
Yeah, it's interesting. I mean, could that potentially be a reason that we're not seeing so much resistance in people because it's an ongoing cycle of evolution? Anyway, thank you.
Yeah, I mean, we do see evidence of resistance, and you kind of showed that in Jerry's talk looking at the colored dots. So we do see evidence of phage resistance in vivo, in our cases.
Thanks.
So I want to thank Dallas, and in the interest of time, we're going to close the session. We didn't have time for a panel, and for all of those in the virtual world, I tried to answer some of your questions and direct you to the different speakers. If you want to, some of us will be able to stay a couple minutes afterwards, but please, I want to thank you for your attendance and thank everyone who was watching this remotely, and continue to have a great conference. Thank you.